My name is Paul Steen, and I am the MiCorps Stream Program Manager. And this is a presentation that should last till about 12:15 that goes over the macroinvertebrate monitoring procedures and the habitat assessment procedures. So, first of all, I think it's important and right to acknowledge how cool aquatic insects are. I do this job because I think macroinvertebrates are the coolest things in the world. And I'm guessing most of you think that maybe it's not, maybe even if it isn't the coolest thing in the world, that at least it's pretty cool. They're diverse and interesting. They just, there's so many different types found in different places that look different from each other. I like the puzzle aspect of it, how some things look so similar, but yet maybe have entirely different biologies or found in different places. So there's just a lot of neat things about aquatic macroinvertebrates, and I think this is why volunteers like doing this project. Throughout this presentation, I may say bugs or macros or benthics or insects or BMIs, but technically when I say that, technically all these things are benthic macroinvertebrates. They are primarily aquatic insects, but there are crustaceans and mollusks and a few other types thrown in there. And the reason we do this work, besides it's just really cool, there's two good reasons. Studying benthics is good science and it's good for volunteers. So just to start on the volunteer side, these are easy sampling techniques, not very technical. It's something that someone can learn with a little bit of practice. Unless you're in a really degraded stream, they're generally going to find significant amounts of them. So they get immediate return on their efforts, right? And it's a unique experience. It's something that most people, it's not something you can accidentally do. It's something that takes intentionality. And when you do it, when you go out and collect these, it's different from other types of activities. We often get comments from volunteers like, I can't believe how many there are down there. And like, I had no idea, those are, if you've been doing this for a while, those are all comments that I know you've heard before. I think there's just a certain sense of wonder that comes about with benthic macroinvertebrate sampling. But so it's good for volunteers. At the same time, it's good science. These stream insects live in these conditions 100% of the time. So they go through the calm periods in a stream. They are there when there is a flood going on. They're there when it's a drought. And if they're not there, that means they can't live in those conditions. So they're indicators of that condition. If you do something like chemistry monitoring, a lot of that depends on just being there at the right time, and maybe you'd pick up a spike of a certain nutrient or chemical that you're looking for, and it's very hit and miss. But benthics are there all the time. So it's a little more reliable than chemical monitoring. That's another reason why it's good for volunteers. And basically the more you find and the more different types you find, the healthier your stream is. What it doesn't do is it doesn't necessarily tell you what is wrong with the stream. If you go to a place and you just aren't finding very much, it will require further investigation to figure out why that stream is low in its macros. It could be sedimentation, habitat loss, chemical or organic pollution, a variety of things that could be degrading the stream, but it at least serves as that initial pass of understanding where your healthy streams are, where they are in between, and where are those degraded streams. So today we're going to go over this outline of a survey design first, followed up by these other aspects of things you need to think about when you collect benthic macroinvertebrates. None of this is hard, but it can be detailed. You know, there's a difference there. This isn't rocket science, and I don't like to pretend that it is. But there's a lot of things to make sure you get things right. There's some details that you need to remember. Okay, so when you're brand new at this and just starting your process of figuring out where you want to sample, the first thing you need to do is figure out where you're going to go. More sites are obviously more better. The more places you can go to, that's great. It gives you a better feel for your water body of interest. But of course, there's money, there's labor, there's time restrictions. We can't do everything that we might want to do. So where do you pick your sites so that it will make the biggest impact and tell you the most? MiCorps requires 6 sites to get that implementation grant. So it's kind of like the baseline. The hope is that as your program grows, as you get more volunteers, that you try to expand that number over time so you can get to more locations and learn more about your watershed. So this is a map of Mill Creek, which is in my watershed, and the brighter blue line, that's the Huron River. So if I was to design a monitoring protocol and pick the sampling sites for just Mill Creek, this is what I would do. You start with a primary site that is basically at the mouth of your system. And the reason you start there is because this would be the site, if this is all you could do, this would be the best place to sample. If there's problems upstream, it should be picked up at this point because all the water flows by it. Obviously, that would leave a huge area that you can't specify, where if you find a problem, you don't know necessarily where the problem is coming from. So it's not good enough, but that's kind of your starting baseline point. And then what I would do is work my way upstream and look for major tributaries that are coming in and try to sample at those mouths as well. So in a perfect world, I'd want to sample at these points next as kind of like my secondary sites, because each of those takes off big chunks of the watershed. There's water flowing, significant tributaries flowing past each of those points. And then what you do is just go further and further up into your watershed and pick out other branches that are draining major areas. So that's what the yellow would be. And then you can go, if it's a big system, you go even further and pick other locations that branch off even further. Now, all of this is a perfect world because the reality is we are constrained in where we can study. So that would be if I had full access to everything. But of course, you have to consider private property. You can't sample where you don't have permission. If your whole area is on public lands, then maybe it's not an issue. Maybe permission isn't an issue, but maybe access, like how do you get your volunteers to that location, could possibly be an issue if there's no roads or crossings nearby. You also have to always consider their safety in terms of you want to avoid particular sections of the river that might be really mucky or really deep. All of this is supposed to be done on wadable streams. So you have to be able to put on waders and walk out there safely. So in a perfect world, your map might look like this. In reality, all those sites are going to be scattered a little bit depending on where you can get permission, where it's safe, where you can get access. So all of those things matter. You might want to first develop a map like this that would be like your best situation, and then actually consider where you can get in and maybe shuffle some of those points around a little bit. Just as an aside, macroinvertebrate monitoring does not require permitting with the state. So you don't have, if you were to do mussel or fish sampling for example, you would need a scientific collector's permit, but not macroinvertebrate monitoring. Another situation that you may have is that you're concerned about a particular branch or a particular area of the watershed. Then, of course, you would locate a site on that particular region of concern. Or you maybe have a landowner who's really interested and wants to be a partner. You know, so all those things kind of play into where your sample sites actually end up. In terms of sampling design, in terms of time of year and frequency, MiCorps groups monitor twice a year for macroinvertebrates. You do it once in the fall, so either September or October. You do it once in the spring, and that has a very wide range depending where you are in the state. Here in Southeast Michigan, we always try to do this sampling in the last week of April or the first week of May, but I know groups in Northern Michigan are often late May or even into early June. Basically, their weather is a month behind South Michigan. So that's just, so you place that date at a time that's appropriate for your position in the state. What you can do is find other groups that are already established that sample these and ask them what they do. You can ask me and I could help you with that if you're trying to pick a particular time of year to do this. One thing you want to do though is pick about a 2-week time frame and always sample in that time frame. So I always sample in the last week of April. And stick with that year after year after year so that the insects continue to be comparable from year to year. Okay, point number 2, let's talk about teams and equipment. I mentioned this in the last presentation, but some of this stuff bears repeating. You need to have a special training for the people who want to be the collectors. So you'll have this team of people, but only one of those people is actually in the river, sometimes two, but they need to have gone through a special practice event in which they actually practice their collecting techniques. And you don't want their first time experience doing that to be an official collection. So go over the procedures, show them what microhabitats mean, which we'll talk about in a little bit. And then this is what I always get. I get someone coming up to me who's new to the organization saying, oh, I've already done this so much. I did it with this organization or that organization, or I'm a professor and I do it professionally and this and that and so on. And my comment to them is, great, we're lucky to have someone like you who is going to help us on this project, but come to our training, so make sure you know our specific sampling constraints and procedures. So yes, people may have done this before, but if they haven't done it with you, they still need to be trained because they don't necessarily know 300 feet, the time constraints, all of that. So you still need to run all that stuff by them. So people will try to get away with not being trained, but don't let them. It's also important that you don't make newcomers sit through long trainings, because being a picker is not difficult. As Jason mentioned, we can have three-year-olds and 4-year-olds who do picking. It's just a matter of being patient and having tweezers, right? So give them a 10 to 15 minute introduction. While on site, you can have someone who's done it before show them what to do, but you don't want to put unneeded barriers for participation because that will lower participation, right? So don't do long trainings for pickers. Okay, so this is just based on my experience. What I have found is that the best team that you can get is somewhere between 4 and 6 people. I've been doing it this past year because of COVID with teams that have been 2 to 3 people, but the longer we've been doing that, the more I decided I don't really like that because I don't think they're bringing it back enough insects to to make for good samples. They're often coming back with like less than 50 or sometimes less than 40. And I just don't think they're spending enough time doing the picking. But if you have a team of 4 to 6, they're gonna come back with the proper amount in the amount of time that we lay out for them. Never send out a single person. Always make sure they're at least in pairs. If you start having huge, gigantic teams, that's up to you. But what I have found is that like a collector can't keep up with them in terms of their sorting. They'll start running out of equipment. They'll start tripping on each other. Some sites, you may not even have much of a bank to set up your stuff. Other sites, maybe you're at a big park and there's room, but so maybe it's possible in sites like that. But in any case, what I have found, really my prime best team that if it was up to me, it would be 5 people, because you would have the trained collector, you would have a leader who stays on the bank, who helps the new people and fills out the paperwork. You would have a collector's assistant. That's someone that you can put in waiters if you have enough waiters, and they can help the collector carry the trays back and forth to the team, so the collector doesn't have to waste their time going back and forth from the bank, especially in a wide creek. That's really useful. And then you'll have 2 people who just do the constant picking. The leader will pick also, and the collector's assistant will also pick. So all combined, you would have 4 people doing the picking with 1 collector. That would be the best situation, in my opinion. In terms of what equipment you get and give to your team, so go to our website, and then under the drop-down streams menu, you'll see stream documents. That's where you can get to all the documents that we talk about, the data sheets and the list of equipment. You know, if you're brand new at this, try visiting different groups. Every group should kind of do things the same way, but they may have little things that they differ on. So seeing different groups in action can help you learn ways to make your procedure work as best as possible for you. This is kind of an aside, but it's important to talk about. So in terms of safety, the collector should not be going off and disappearing from the team entirely. So keep your collector in sight. Make sure your collector is moving slowly and cautiously. People who have experienced walking in streams, like anglers, would be like really good collectors typically. Beware of things that they can be tripping on, things that are dangerous in urban streams. You may have rebar sticking out, for example. If your streams are at all deep, I'd say anything starting to get above the knees, and if it's decently fast water, you should consider giving them life jackets to go out in, and not just the waders. Certainly when it gets over their waist, you're at a point in which life jackets probably should be mandatory. And then there's things that you should be doing in terms of safety as a coordinator. You need to be obtaining those permission from private landowners. Let them know when you are coming so that they don't have a bunch of strangers showing up at their property without them knowing. Contact your health department, contact Eagle for specific streams. So some things you can be thinking about is some streams may have E. coli issues, and you want to be careful with people washing their hands, not going in without waiters. Some streams have PFAS nowadays. There's plenty of endangered species out there that we want to make. If you go to a location that has New Zealand mud snails, your team should know that they have New Zealand mud snails, so they can be extra cautious about not transferring them. Send teams out with first aid kits, and then have volunteers sign waivers saying they will not sue you. I do have some, I can provide language for that if people email me and ask, but that's the kind of thing that you should then run by your own legal counsel to make sure it will work for your organization. Okay, now we are at collection, and this was the point. If we were together in the stream, we would just talk about this right on shore and then go in and do it, but we can't do that. So I'm just going to kind of like lay out the gist of it. There's a couple numbers that you need to keep in mind. Your collector, when they go in the water, they should be doing it 35 to 45 minutes. So that's the length of, or the total effort for a 300-foot stream section. There's a reason that you have a 10-minute window there. That's because some streams are harder than others to walk in. You can see in this picture, this person's going to have to be crawling over fallen logs, so it's just going to slow them down. If the collector has to do a lot of going back and forth to the bank, that will slow them down. So it's really your discretion whether they're going to go that full 45 minutes, or if it's an easy stream, they're just zipping along, maybe 35 minutes is enough to cover 300 feet. So that, just to give a window for your discretion. Always have your collector work upstream, always headed toward clear water so they can see what they're doing. And then as they're doing this, they are looking at the stream and considering all the microhabitats of the stream ecosystem. So the way water flows around obstructions and around bends creates unique environments for the different macroinvertebrates to live in. You're going to find different macroinvertebrates living on the stream margins in pockets of muck than you are in the middle of the stream where the water is flowing fast. So to collect as wide of a diversity of the different taxa as possible, you have to get to all of these places. Some streams will have them all, some streams may only be riffles and pools, but in any case, you instruct your collectors, like, this is where their mind should be. When they're collecting, they're looking for different habitats because they can't legitimately sample every square foot of the 300-foot stream section. They are going to be popping around back and forth, working their way upstream, nailing these different microhabitats as they go. You know, you don't need to sample an entire 300-foot riffle. You would do 5 attempts at a riffle and then move on to the banks and do things over in the banks. So at all of those microhabitats, they're just using their net to pull things out. Of course, you get more debris than you do insects, but that's not the collector's job to figure that out. A lot of times you'll see collectors take a net of stuff and then look in it and they don't see anything moving and they think they didn't catch anything. And maybe they'll dump it back, but they should not do that. That's not their job. Their job is to collect the stuff and give it to the rest of the team. And the rest of the team's job is to sort through all that debris. So they should never be dumping stuff back in after they take a net of it. They should be bringing it over to the team on the bank. You have a couple options here. You can have a team sitting right there on the bank with trays and tweezers and doing the picking. Some groups like to just put all that debris into a bucket and then like bring it somewhere else, like under a picnic shelter or something, and do the picking there. So that's kind of your choice. I prefer the team picking on the bank immediately. I think it's a little more efficient. Now muck is miserable. I say minimize muck mass. So if you have mucky areas, don't do more than a few samples at the muck. Just give 1 or 2 nets of muck to your team because it's so hard to sort through heavy silt. And whatever you can do as a collector to like minimize the amount of muck you give your team, that's good. And chances are you're only going to find a few things in it anyway. It's just not a very biologically active habitat. So keep that in mind. Okay, pickers. Direct your pickers, the people on the bank, to, so they're finding moving things in their trays, and they are putting them into small jars of ethanol or isopropanol, a preservative. They should be striving for at least 60 creatures, though 100 is preferable. We don't usually have them count unless you want them to, but especially with a team of any size, it's going to be hard. They'll be going fast and it will be hard to keep an accurate count. So You just try to have them have a general sense of how many they're finding. And your pickers can go up to 60 minutes. Your collectors stop after 45, but your pickers can keep on going because they'll still have trays of stuff that they have to go through. So give them up to 60 minutes. If you're only picking with like 2 people, you can expand that amount of time to make sure that you get up to the over 100 number. Yeah, and just as an aside, if you can have someone with experience that I always call the team leader, staying on the bank, working with the new people, that will be very helpful to help them learn how to do the picking. So that's just a useful job to give somebody. Your pickers shouldn't be preferring one type of creature over another. Have them look for small things and big things. And if they see them, they keep them. An optional thing is if they pass over about 150 and they have 3 jars and they're all full, at that point you can have them stop. And there's certain things that that you should not be keeping. So any clam, mussel, snail, fish, or crayfish, those are things that you want to note on your data sheet right away. But then you don't put those in your alcohol, you release those. An exception would be if teach them what New Zealand mud snails are, and if they think they have found the New Zealand mud snail, they could bring those back and show them to you so that you know if they're at a site or not. If you feel like you've found them, you can either get in touch with an eagle biologist, or you can email me, send me a picture. I'll help you find the right person with the state government that you can report your find. Obviously, if it's already a well-established site with New Zealand mud snails and Eagle knows about it, I don't think you have to tell them again. Following collection, there's decontamination. So the exact procedures of decontamination can reside with you, but the basic gist is that you are getting rid of debris off your equipment and you are allowing either a dilute bleach or other chemical like 409 to remain in contact 10 minutes with your equipment and killing off anything that may be hanging on. You know, and the places that you really concentrate are like things that have had direct contact with debris. So wader boot treads would be probably the hugest, as well as the net head itself. Then everything else, you can use the water at that site to do the rinsing, and then you finish it up with spray bottles of the bleach and 409, and then you can use tap water to rinse everything off at the end and dry it, and you should be good to go. Here's a particular website you're welcome to follow. That's with my organization, how we do it, and we have a DEQ Eagle approved video that we produced a few years ago that talks about how to do decontamination. Okay, now we're at the identification phase. Tamara at 1 o'clock is actually going to go through pictures of the insects and how to do the tell them apart. This is more like the logistics of identification right here. So it's up to you whether you want to do identification immediately after collection or hold it as a different event sometime in the future. Have your volunteers look at the bugs, try to figure out what they are, whether they collect, even in the moment they're collecting them, or later on during your event, but that's never the official ID. So the official ID and what's actually being written on your data sheets has to be done by an expert ID, someone who has some level of experience at distinguishing these. A lot of times you can get volunteers with this experience to come and help you. Maybe you have that experience, or you can build up to that level of experience. And I leave it up to each group to determine who those ID experts are. And of course, it's in your best interest to get the best data possible to be picky about who that ID expert is. So don't do the official ID in the field. Don't do the official, oh, I have a total typo. Oh yeah, you should not, right. Yeah, I thought I had a typo there, but it says you should not. You should not do the official ID in the field. You should not do the official ID on live creatures, not in MiCorps. In MiCorps, we put insects in preservative and keep them back because we're saving them, these samples, for 5 years at a minimum to maintain our kind of our scientific integrity. If anyone asks about your samples, you have them on hand and you can show them that your identifications are correct. Don't just save a few insects of each type, especially because it would probably be your volunteers who do this out in the field as they are collecting. If they're only saving a few of each type, you are asking your volunteers to make a judgment call on identification that they may not be trained for. You know, something that would be consistently messed up would be free-living caddisflies. There's 4 different types of free-living caddisflies. They all look the same. So having them just save a few of each type is going to result in errors. So they shouldn't be making that judgment call. They should be keeping things and letting experts look at it. Okay, so now that you have done your identification, you'll be scoring your insects. Now we're going to use a method that's called the Hilsenhoff IBI, the Index of Biotic Integrity. And what the Hilsenhoff does is measure sensitivity to organic pollution. So organic pollution is, it's wastewater, it's fertilizers, nutrients, pesticides. These are substances that are strongly connected to oxygen levels. As those substances decay in systems, it takes up oxygen. And there's been a variety of studies that have given particular numbers to different taxa that show how much that affects them, how easy it is for them to survive in an area that has lots of wastewater, for example. Typically, so that uses terms like sensitive and tolerant. Something that's tolerant can live in a stream that has lots of those substances present. Something that's sensitive disappears when you have any amount of that sort of pollution. Secondarily, the numbers are also related to the habitat quality and the flow. That's because areas with organic pollution will typically be more degraded in general in terms of habitat, because they'll have some kind of stronger connection to land use or development impacts. So streams that have a lot more organic pollution are also going to typically have more fine sediment and bank erosion and riparian vegetation issues and less in-stream habitat, perhaps water flows that change really fast after rainstorms. So altogether, all of that kind of is incorporated in this Hilsenhoff number. And this is what your data sheet looks like. That 4th column says sensitivity rating. So that's that Hilsenhoff number. So this is rated from most sensitive going down to most tolerant, like a leech and a ligochaete worms are going to be the things that can live anywhere, no matter what the quality of your stream is. Well, Helgramite and club-tailed dragonfly and stoneflies, those are the best in terms of water quality. If you have a lot of those, it means you have a very high quality stream. So I'm going to just walk you through this data sheet. The first thing you do is you'll have counts for each of these taxa groups after you do the identification. So you'll just add those up. Down here you can see we have 75 in this particular sample. Now there's an abundance check here. If your volunteers come back and they have less than 30 insects, that is generally considered a really bad sample. There could be a couple things going on. It could be the stream is completely flooded because of maybe high water and they can't even really get in the stream to do the sampling, in which case that's your job as a coordinator to reject that sample. And it doesn't officially go into your long-term trend database. That's your job to kind of make those calls. I always keep those outliers to show, what can happen under certain conditions, but it shouldn't represent the normal water quality rating of the site. In any case, if they do come back and there's no particular reason for rejecting the sample, like a flood, and they only can find 30 insects, that's a really bad sample. It automatically gets scored as a 10, which is very poor. This is because the Hilsenhoff index really relies on having an abundance of about 100. And we say about 60, you start to it starts to being okay in terms of the abundance that you find. An extreme example would be if you go to a location, you only find one stonefly, maybe it washed there from upstream, and that's all you find in your whole sample. Well, if you score this as normal, you would end up with an excellent rating with 1.3. And that's because under low abundance situations, the scoring system just doesn't work anymore. So we have to have a couple of specific breakpoints that tell you when you have enough to do the scoring properly. So less than 30 is considered 10. If you have less than 60, it's just given a 7. And if you can get above 60, there's no penalty anymore, and you can do the scoring as normal. You always try to strive to get about 100 to properly do the Hilsenhoff IBI. And greater than 100 is totally fine too. More bugs won't change the score. Okay, so if you have enough, you proceed with this and you're multiplying across. So I would take 35 caddisflies and I'm multiplying it by the sensitivity rating. And that is going to get me 112 for this line. And you want to do that for each of your lines that you have found insects for. And then you add that column up. So in this case, adding that column up, I get to 335.5. Then this column over here, the water quality rating, is going to be 335.5 divided by 75, getting me this number, which is your final rating for the score, or for the site. It will put you at, right on the boundary of this very good category. So that's your final rating for that site. In terms of eagle biologists, they go by that score, but they'll also they'll want to see what you actually found too. So when we share our information, we try to both give a breakdown of the taxa as well as the final score. If you are a group that has the expertise to do family level identification, there is a data sheet that exists for you. It looks like this. You would be identifying to each of these families, and each of these families has its own tolerance value. Some of these families I couldn't find numbers for. That will be a work in progress, but they may not have been studied with the Hilsenhoff IBI, like these beetles, for example. So there's variations allowed in how people do things. And we allow for that. There's a range of preferences and you can make some choices. For example, I've already given you my opinion that pickers shouldn't be overtrained, but some groups train everybody. That is ultimately your choice. That doesn't affect the data, so that's fine. Some groups like using 2 collectors in their stream with 2 people with nets instead of 1 collector. That's okay. Just your overall time frame should still be 35 to 45 minutes. You'll just add up their individual times. It's okay to use slightly different equipment. Like if you wanted to use kick screens that are a piece of equipment that work really well in riffle areas. That's fine. It doesn't change the, you know, the types of bugs. It just kind of facilitates finding them, makes it a little easier. It's okay to use different sizes of white trays. My group uses these little standard white trays, just because we have so many of them at this point. But if you're starting out, there's these monster white trays that you can get that go under refrigerators. And I actually like those more. I think they're a lot easier to use, but then people need big cars to transport them. So you can consider looking at those and thinking if they would work for you. My teams, for my group that I run, we pick on the bank, but some people put everything in a bucket and they pick it later, and that's okay. I just think that's a little harder. You might miss things that way. When ID happens, I talked about that already. Collect everything on one day versus a 2-week time period. So I haven't mentioned that. My organization sends everybody out on the same day. But if you wanted, you could stretch that out and have people, a team go here, a team go there, kind of depending on their schedule. And that's an acceptable way to do it. But the goal there is you want everything sampled within 2 weeks. So you don't start seeing drastic changes from the people who did it at the beginning versus the people who did it at the end in terms of insect biology, because the insects are always emerging and always hatching and changing the community in the river. Some things are a must in terms of MiCorps procedures. Like you have to do these things. You have to train your collectors ahead of time. You have to follow our numbers in terms of collection effort. Don't pick for more than an hour unless you just have a tiny team that needs more time to get to that 100 bug mark. Collect in that 2-week window that's similar year after year. Don't do your official IDs out in the field. It should be done in a controlled environment with dead specimens. You need to have ID experts to do this. And you want to keep the specimens for at least 5 years. And then QAPs, which are your quality assurance plans that these established groups will have. And me visiting the groups and side-by-sides are kind of MiCorps' way to ensure that generally between all the groups, we're all doing approximately the same procedures. And that's a way of maintaining our standing with the state and with other organizations who may use our data. So it's an important thing that we're all generally doing the same thing. Whew. Okay, so that's actually all of the macro stuff. And I'm about on target. I don't have as much to say about habitat assessments. But I'm gonna look at this one question. Joyce is asking, does isopropyl preserve as well as ethanol? Does it need to be replaced over time to keep preservation properties? Okay, so this is my experience with that. Ethanol is more expensive, like significantly more expensive, I believe. But it is a better preservative. It will last for years and years and years, as long as it doesn't evaporate. Isopropyl will kill things and it will preserve things, but it makes insects really brittle. So if you have insects saved in isopropyl alcohol for years, they may start breaking apart over time. Now, if you don't have any intention of keeping your collection more than 5 years, isopropyl is probably fine, and it is cheaper. It's rubbing alcohol. It'll be easier to procure. Ethanol is kind of a pain to buy. The cheapest ethanol I have found is going on Amazon, and you can get big jugs shipped to you, but the shipping of ethanol costs a fortune because it's a hazardous substance. So yeah, hopefully all that info can help you make a decision. Just take a drink here. Any other questions on the bug stuff? And give us a little bit of time to talk about the habitat assessments. How does the new water quality rating compare to the previous stream score? We have 8 years of previous data. How do we incorporate and compare scores calculated with a different method? Yeah, I'll talk about that. But Sally says, I suggest contacting local universities to assist in ordering ethanol. That's a great suggestion, Sally. Thank you. Yep. Local universities have much easier time of getting ethanol because they order it in such mass quantities. Kelly, your question is, I spent about 20 minutes talking about that earlier in the day. So if you missed that presentation, that was at 9, that whole presentation will be available in a week or so as after I process all these videos, and I'll post it online. So you'll want to watch that presentation to get the answer to that question. Heather asks, Paul, can you confirm that the collectors can put everything in a bucket and then join the pickers to do the hour-long picking event? Yes, I can confirm that's a fine procedure to use, Heather. It's okay if you just send out the collectors to do the picking. And then you pick at a later point. Yeah, that's approved. Okay, so let's talk about habitat assessment. Oh, and all this, by the way, if you have more questions, like just email me. I'll put that. Or someone, maybe Tamara or Joe, could put my email in chat. You know, ask me questions, and I will get back to you as soon as I can. And I love it. I love starting those dialogues with people. So, okay, so the habitat assessment. What is it? So the habitat assessment is a complement study to the macroinvertebrate monitoring. So the macroinvertebrate monitoring, through that you find out what insects you have. The habitat assessment helps you understand why you're finding them at that site and why their populations may be changing. So it starts to get a little bit at this why issue, which is why we have groups do both of these things. Now, a habitat assessment is a qualitative assessment, not quantitative. So you're not going to be going out there and measuring a million things with a tape measure, but you're going out there with a team and answering a series of questions on a form that give you a good general sense of the habitat at that location. And so I'm going to kind of walk us through this process, though it's really the kind of thing you have to see in person. So the people who are, there's one group that has an implementation grant and will be starting to do these. So Nancy, your group, I'll actually plan on, when I do my side-by-side with you, I'll go out and actually do one of these studies with you and walk through it with you. But for right now, I'm just gonna kind of walk through the logistics of how the habitat assessment works. Okay, so first of all, here's once again our website and where you can get these documents. You can get the habitat stream procedure from this point, as well as that stream habitat data sheet from this point. And the habitat procedures go into pretty high level of detail. So it's definitely something you want to read a couple of times if you're going to do this. So they're not hard to do, but they can be initially confusing until you get the hang of how they work. So because of that, it's never something that you just give to new volunteers. It's going to have to be someone who's been trained in it, that maybe you can do this as part of your collector training and show people how to do a habitat assessment. Or you can actually go out with the teams yourself and do it with them. And one of the reasons why that's possible is because you don't have to do habitat assessments constantly. It's something that you have to do once during the course of a 2-year grant. And then after that, you kind of do it as needed. It can, for my stream sites, for example, we only do it once every 5 years because habitat just doesn't change as fast as the insects. So you can just do it, you can do it more infrequently, which means that it frees up you being able to actually go out with your volunteers to conduct these. Yep, so how often do it? Certainly if you have a grant, you're on, you're obligated to do it at least one time at each of your sample sites. If you're not funded by an implementation grant, there's no hard rule. I just recommend every 5 years. And if you feel like your stream is changing actively, like there's things happening there that are changing the habitat, maybe you do want to do it every year. So that's your decision. When to do it. So you can give your teams the option of conducting a habitat assessment after collecting macroinvertebrates, or this is my preference actually, hold the habitat assessment at a different time of year and treat it as a different event. Give them 2 or 3 sites to go to that are close by, and then just have them hit them out in a couple hours. Summer is a good time to do this because we're not actively collecting macroinvertebrates in the summer, and you also seeing the vegetation growing on the banks is a useful part of the habitat assessment. So I suggest summer. How to conduct this. All right. So first of all, you need to be at the site for some time in order to answer the questions. That's why it is a good complement to a macroinvertebrate study, because the team has already been there for a full hour. So they've had a chance to explore the stream a little bit and think about these habitat issues prior to answering the questions. If you go out as a separate event, spend 15 minutes exploring, walking the stream and poking around before you try answering these questions, okay? So after your initial exploration of the stream is done, you'll stand in the stream or next to the stream, and one person's gonna hold the data sheet, and you just kind of walk through the data sheet together. The group, by consensus, rates different aspects of the habitat. I'll show you an example of this so you know what I'm talking about, but everyone's allowed to chime in and give their opinions about the habitat. And if together, if you can't agree on an answer, you take averages when you're talking about numbers, or you record comments to get people's opinions in this. Like I said, this is very qualitative and not quantitative. Here's an example. This is what I guess would be a more quantitative part of it. If you're talking about the riparian width of a stream, you have to rate each bank from 0 to 10, where 0 would be a terrible score and 10 would be an amazing riparian zone. So the person who's holding the data sheet kind of reads through these options, or the team can kind of gather around and read this. And together you decide for each bank what the score should be for this particular stream. So for example, you're at a stream that there is maybe a scattered tree here or there, but mostly it's just mowed lawn. Like what if you're at a park and it's just mowed lawn everywhere? That's going to be a pretty poor riparian zone. You would give that somewhere between a 0 and a 2, and your team would talk about what that exact number should be and come to consensus. Maybe one person says this should be a 0, and another person says I've seen worse, I'd give it a 3. Then they can argue it out and come to a consensus, or you can take an average of their opinions. Here would be this is another example of the type of thing you would go over on a habitat assessment. Sources of degradation. So you're standing in the stream, you're looking around. What are ways in which the stream has been affected by human impact? Perhaps you are, your stream flows right through an agricultural field. You're right there, you see it. And the team may vote, this gets a high impact from crop-related sources, for example. Now, this always comes up. Remind the team it's what they can see from this location. You can't assume that they have this high level of knowledge about the entire watershed and what might be impacting it. It's just what they can see. Because going beyond what they can see then relies on outside knowledge that you can't control. So it's just what they can see. Right, so here's some other parameters that your team is going to be going over as you are out there. Stream size, flow, presence of foam or oil or trash, the condition of the stream banks, how erosive are the streams. That's another one that you rate from 1 to 10. different plants in the plant community, how many invasives are here versus native plants, size of the riparian zone, sources of degradation, which is this, you saw half of the table, but it's a long table that lists a whole variety of things that can be affecting the stream. And then an optional pebble count. This would be a way to get more qualitative information. The procedures cover this, but basically you can stretch transects across your stream and at even intervals record if it's sand, cobble, gravel, boulders. And with that, you're able to generate a percent substrate for the stream. And that can be interesting. That's a quantitative measurement. And you can see how that changes over time. Might be something you want to do if you're worried about your stream collecting sand over time, for example. So normally I would go through all of this together standing out in the stream, but we can't do that on Zoom. Really the best way to learn this stuff is to be just out there looking at things, looking at the data sheet. And that, I mean, that's really the best way. So Nancy, with your group, will go ahead and do that. All the other groups have already learned how to do this, and you're just here for a reminder. Or if you're a beginning group, once you get an implementation grant and get me out there with you, we'll do this in person in the future. So